time, I'm going to start this first session of the day, in which we have 7 talks that I'm sure they are going to be amazing from researchers all over the world. They are researchers of excellence in the area, who I thank for accepting the invitation to be here today. The talks are going to be between 10 minutes and maximum of 15 minutes. Say that if this time is reached, then there will be no time for questions, and then the questions will be held at the end of the session in a general discussion time that we have at the end of each one of the sections. I will also tell the speakers around 15 minutes if they reach that time, that the time is getting there. For you, please, to finish your talks, and then we can stay with the time for the rest of the speaker of the session. So thank you again to all the speakers, and I will introduce the first one of today and this session, which is Professor Phil Hugenholtz from University of Queensland, Brisbane in Australia, with the talk Finding the Needles in the Haystack, Data-Driven Selection of Therapeutic Candidates from the Human Gut Microbiome. So thank you very much, Phil, for accepting this invitation and for your talk. And the virtual stage is now yours.
Thanks, Beatrice, and thanks, Gloria, for the invitation again. Welcome, everybody from Australia. You're also welcome to visit here. I'll try and stay on time. Can everybody see my slides?
Yes.
Yes.
Yeah. Okay, great. All right then. Okay, so I'm going to talk about data-driven selection of therapeutic candidates from the human gut microbiome, and I often compare this to identifying needles in a haystack. This is not actually work done through my university, with my university hat on. This is a company that I co-founded with Gene Tyson in 2018. And it has the mission I'll call Microbiome, which has the mission of doing precision microbiome testing and therapeutic drug discovery. So I don't really need to tell this audience that there's been a big shift in the way we think about microorganisms in relation to humans. In the old days, we thought the only good bug was a dead bug. And these days, we think of microbes as an essential part of our livelihood. So this has the implicit assumption or presumption that some microorganisms are explicitly good bugs and some are explicitly bad. We know it's a bit more nuanced than that. It's context dependent, but we're basing the idea that in amongst all of the organisms that inhabit our bodies, there are some that are particularly good, and those are the needles that we're trying to find in the haystack. This is not a new area where people have tried to take gut bacteria and use them as therapeutics. And so if you look in recent times, about 2005 to 2015, you would just do whole transfer, fecal microbiota transplant. So you would take fecal material from a healthy donor, and you would transfer that into a patient with a disease, in the idea that you replace bad microbiome with a good microbiome. This is not a new idea at all. They were doing it a couple of thousand years ago in China. A bit more sophisticated is really front and center in 2015 to 2020, where you do random screening of culture collection. So rather than just taking a gamish of microorganisms that in whole stool, which carries some risk with it if you've got pathogens in there, you instead have a culture collection of bacteria, not necessarily just gut bacteria, and then you screen them for some attribute that would be useful as a therapeutic drug. And most recently, and this is what Microbiome is focused on doing, where you actually use data that you obtain from a large cohort of people to rationally discover organisms. So rather than just randomly looking for them, you have a directed discovery, and I'll talk about that now in a little bit more detail. So the idea is we've created this platform where we take a large population that has been tested, and the idea there is then to split the profiles, the gut microbiome, and this is metagenomics using shotgun sequencing, so you're getting access to all of the organisms that are in the gut, and you identify a healthy cohort, and we do this through questionnaires, so we know that this is the healthy fraction of our group. And then you have some sort of disease condition, and you get the gut profiles of those people, and then you compare them using statistics and some AI in order to identify particular microorganisms that are overrepresented in the healthy humans relative to the diseased humans. The idea being that those organisms are depleted in the diseased individuals, and therefore they are contributing to the health status that is missing in the diseased people. And so then we identify those ones from the large data set, and then we isolate them, and then we test them out to see if they have some useful biotherapeutic properties. So for this, you really need big metagenomic data sets to get the statistical power. And so Microba has spent a lot of time and energy building, first of all, getting a consumer microbiome collection kit where you can just get that ordered, send out to your place, take a bit of fecal material, and then it gets sequenced and analyzed in a very reproducible way. And then we built this database of genomes and genes. And at the last, we spent quite a bit of time on both the lab side of it and on the bioinformatics side. And at the last GloMiNe meeting, I talked about one study where we evaluated different ways of preserving the fecal microbiome profile. So we also have in the bioinformatics, we have a community profiler which we built, which is we think is pretty accurate, and a very large reference database. Just to give you one example of the sort of thing we've been focusing on in my group at the university was to refine the classification, because the classification is important to get to be able to see biological signals. So this is an example of one well-known gut microorganism, Faecalibacterium praunitzkii. And so if you look at the existing taxonomy, it's just identified as one species. But if we take it apart at the genome level, we see there are at least 20 distinct species. And we know that these different species have different properties. For instance, Praunitzkii C is involved with atopic dermatitis, and Praunitzkii G is often associated with a healthy microbiome. So it's really important that you can get that level of resolution. So a flagship study that we focused on was inflammatory bowel disease. It's a chronic disease which is really quite debilitating. It affects more than 6 million people globally, 1 in 250 people on average. It's mainly characterized by inflammation. There's two types: Crohn's disease, which is the nastier one, to be honest, and ulcerative colitis, which is more common, and it's just about where the inflammation is localized in the gut. So we took our customer profiles and supplemented that with public data sets, ran them through our pipeline to identify high confidence leads. So this is we got about 20 species where we saw statistically significant overrepresentation of species in the healthy cohort relative to the IBD cohort. So it's just important to note that recent estimates suggest that only 30% of the species in the gut microbiome have been cultivated. So that means if you're just doing a random screen of the library, you're missing a lot of the potential therapeutic organisms. And so that was reflected in our therapeutic leads, that about half of them or more were actually from this uncultivated fraction here. So for those ones that haven't been isolated before, we used so-called genome-directed isolation. So when we do metagenomics, we get back the DNA blueprints of the organisms. So from that, we can do a metabolic reconstruction and work out ways that we may be able to isolate the organism from a background of other organisms. And so the organism that I'm going to focus on is a thing called MAP315, which was one of our top, I think, in the top 5 leads. And first of all, we screened our donors to find a donor with a relatively high relative abundance of our target organism. And then we worked out what conditions would favor the growth of this particular target. And then we reiterated this a few times, and we were able to isolate for the first time this particular species from the gut. This is I want to just make here the point that it's really important to have this resolution. I think this figure really drives it home. So this is a genome-based phylogenetic tree. Each tip represents a different species. Here is our guy that we're interested in. And the little line, the distance between the line, is the indicator of the relative abundance in the healthy versus the diseased state. And so you can see that's got the longest line here, and that's why it's one of our leads. But it's really important to note that neighboring species in the same genus does not have that profile. So not a therapeutic lead. So it's really important to have that species or even subspecies resolution in order to get the right organism. So we took MAP315 and we put it into a mouse model of ulcerative colitis. Basically, you give these mice a bit of detergent, which then brings on conditions that mimic colitis. And then we added in MAP315, and we looked to see if it had an effect on a number of different fronts. So we looked at inflammation, and so we can see that this is the vehicle, which is the control, and then this is with the after you've added the induced the profile. So high score means a lot of inflammation, and you can see that there's a statistical drop in the inflammation when we add MAP315. Prednisone is currently the standard for treating people with IBD. It's not suitable for long term, and it has some bad side effects. But you can see that the MAP315 has properties that are look very similar to the prednisone. So that was good. We could show it in a mouse model that we were getting improvements. We also could show through a cell migration test where you look for the speed that cells migrate, and this is an indicator of wound repair, that when you added the supernatant from a MAP315 culture to those epithelial cells, human epithelial cells, that you increase the cell migration, which indicates that it's also contributing to wound repair directly. And we could see that MAP315 supports gut barrier integrity, and this is by modulating the tight junctions between the epithelial cells. And so Z01 or Z01 is a protein expressed that is expressed when to induce tight junctions. And we can see that by adding the MAP315 supernatant or exudate, that we could recover, we could get good tight junctions with Z01 that were interfered when we added interferon gamma, which interferes with the function of that protein. And we also could directly show that MAP315 suppresses interleukin-6, which is a cytokine that mediates the pro-inflammatory pathway STAT3. And so we can see here, for instance, that the STAT3 activity is significantly suppressed by the addition of the MAP315. So I just wanted to give a quick flavor that the interesting thing is that this organism is working on a number of fronts. And so I think that's a real benefit of using a live biotherapeutic, because a live organism has multiple functions that can contribute to the therapeutic effect rather than, say, a bioactive. The next thing we did after we had found that it had some therapeutic potential is we wanted to see if we could grow it in large amounts, because you need to do that if you're going to manufacture it. So we did an in silico assessment of the organism. So we were looking, did it have antibiotic resistance? Was it carrying phages? These are no-nos for if you want to grow up an organism, and it passed that well. It had excellent safety characteristics in silico. Then we grew up the organism and played with the media and found that if we removed out animal-derived components of the growth media and replaced them with equivalents from plants, that we could really increase the growth conditions of the speed of growth of the organism. So yeah, excellent growth in animal component-free media. So at this point, we partnered with Bacteria, which is a company specializing in scale-up of live biotherapeutics of microbial cultures, and that took a while, but it went well, and we've been able to now put the organism through a human phase 1b study, which is basically a safety trial, and I'm happy to say that everybody survived. I shouldn't say it like that. It did really well. It went through without any trouble, which you would expect because it's an organism which is derived from healthy humans. And just for fun, this is what MAP315 looks like if you grow it up in large amounts and lyophilize it. And so this goes into the capsules that then hopefully we can all take at some point, because my expectation is that this organism is indicated with quite a few as a healthy organism in general. So it could actually be just a good organism to take in general. So just in summary then, using this data-driven process, we were able to go in about 36 months, which sounds like a long time, 3 years, from using the data-directed approach to isolating the leads and then testing them out. And now we have this lead candidate that's being manufactured for human trials. And just for comparison, if you do just screening of a library, a culture collection, that usually takes in the order of 5 years, and there's no guarantees of success. No, not to say that there's guarantee of success here either. Okay, that's it. Hopefully I did that all within time. These are the fine folks at Microbiome. That's Jean, the co-founder. A lot of the work done here was by Porrick O'Queeve and Lutz Kraus, the chief technology officer and one of our lab heads. And that's it. Thank you very much for your attention.
Thank you so much, Phil, for such an amazing talk.